and welcome to this explainer. Today, we're totally demystifying the S-PrediXcan and Metascan pipeline. If you've ever wanted a clear, highly structured way to predict gene expression directly from summary-level GWAS data, you are in exactly the right place. We're going to break this entire complex process down step by step so you can confidently apply it to your own bioinformatics research. Okay, let's dive into this. Here is our roadmap for today. We'll start by breaking GWAS limits, prep our environment and data, harmonize genomic coordinates, impute the GWAS results, predict gene expression, and finally interpret those results. All right, step 1, breaking GWAS limits. Let's move beyond the genome. So how do we actually move past the limitations of GWAS? I mean, we all know Genome-Wide Association Studies are, quite frankly, incredible tools, but they have a huge bottleneck, right? They really only explain phenotypes at the genomic level. And let's be real, in most real-world scenarios, we just don't have the individual level genotype and phenotype data we need to push that analysis deeper into the transcriptome. And this brilliantly illustrates how the PrediXcan and MetaXcan workflow completely bypasses that roadblock. Instead of needing that massive, almost impossible to get individual level data, these tools let us predict target genes and tissue-specific expression using literally just GWAS summary statistics. It's basically a bridge. It connects a genetic variant directly to a physical trait by predicting the intermediate step, the transcriptome. Moving on to step 2, prepping your environment and data. Getting set up for success. Okay, we've got 3 fundamental setup steps to knock out. First, we download and configure MetaXcan using Conda. Then we set up our dedicated GWAS imputation environment. And third, we download our sample data to actually test the workflow. Just a quick heads up here though, running this kind of transcript-wide analysis is heavy stuff. Make sure your computer has some solid specs because it's going to need the processing power. Now, I need to emphasize an absolutely crucial point here. Strict version control, it's not optional. When you're installing your packages via Conda, you absolutely must specify these exact versions: Pandas 0.25.3, SciPy 1.4.1, and NumPy 1.18.1. Locking these in is mandatory. If you don't, you are guaranteed to run into those deeply frustrating version conflict errors later down the line. We've all been there, it's a nightmare. So trust me on this, lock the versions in. All right, on to step 3. Let's harmonize those genomic coordinates and align our data maps. Now, what's really interesting about this slide is seeing that translation happen right in front of us. Look at the raw GWAS data on the left. You'll notice it's mapped using the older HG19 coordinate system, but MetaXcan's latest models actually require the newer HG38 build. So by running our bash script, all that data on the left is cleanly converted to the updated HG38 standard you see on the right. It seamlessly updates the chromosome positions and makes sure our reference alleles match up perfectly. Next up, step 4. Inputting GWAS results, or basically filling in the blanks. To really grasp the scale of the imputation process, we need to talk about what I like to call the 220 cycle marathon. Your Python script doesn't just run once. It systematically cycles through all 22 autosomes, and for every single chromosome, it processes 10 distinct subbatches. Now, doing that manually 220 times would be, well, soul-crushing. So we use bash loops to completely automate the generation and submission of these scripts. And if you've set those bash loops up correctly, this is exactly what you want to see. This terminal output is your visual confirmation that the script is successfully chewing through your target regions, batch by batch. Once all 220 cycles wrap up, you just run a final post-process script to seamlessly merge all those individual batches into one massive, cohesive, imputed file. Which brings us to step 5, predicting gene expression, the real climax of the pipeline. For this stage, you've basically got 2 core scripts in your toolkit, and it's super important to know the difference. S-PrediXcan is your precision tool. It targets a single, specific tissue using a dedicated database model, like, say, predicting expression solely in whole blood. S-PrediXcan, on the other hand, goes way broader. It lets you run predictions across multiple tissues simultaneously, which is amazing for finding cross-tissue associations. When you execute your script, we're using S-PrediXcan here, your terminal is going to start tracking the MetaXcan association process. You'll see it climb from 10%, ticking all the way up, and honestly, watching it finally hit that 100% completion mark is just the best feeling. It's your definitive visual confirmation that the heavy computations are totally done. Finally, step 6, interpreting the results. Time to decipher the data. Let's move to the next part and see how this builds into our final output. The script spits out this comprehensive CSV file. And yeah, I know, it looks a bit overwhelming at first glance, kind of like looking at the matrix, but this is literally where all our hard-earned insights live. We just need to know which columns actually matter. So the crucial point is knowing how to methodically deconstruct this wall of text. We turn that raw data into readable biological metrics by zeroing in on a few key columns. Gene name is straightforward. That's your standard Hugo gene identifier. The Z-score tells us both the direction and the strength of the association. Then you have the p-value, which flags the statistical significance, and pred-perf-R2, which basically shows us how well our tissue model correlated to the actual measured transcriptome. Hold your horses, though. Before we start declaring a major biological breakthrough, we absolutely have to apply the Bonferroni correction. Think about it. We're testing thousands of genes simultaneously, so a standard p-value just isn't strict enough to rule out false positives. To get our new ultra-strict threshold, we divide 0.05 by the total number of genes multiplied by the number of tissues analyzed. For a result to be considered truly biologically meaningful, its p-value must cross this highly specific line. And there you have it, folks, the whole shebang. A complete start-to-finish walkthrough of the S-PrediXcan and MetaXcan workflow. You now have this incredibly powerful, highly structured tool to predict gene expression using just summary statistics. So, I'm going to leave you with this thought. Which tissues or complex traits will you apply this TWAS pipeline to in your own future work?